Good afternoon, everyone. My name is Hameme Echampuan. My topic is all about the equipment and technique. Okay, let's start about my topic. The first is using the hand lens. To examine the surface of a wood sample, hold the lens close to the eye, then bring sample towards the lens until the surface comes into focus. Focus is maintained by patting and hands together and bracing them against the cheek, just like in the picture. To use the hand lens, hold it close to your dominant eye, then the object you want to examine closer to the lens until it comes into focus. Essentially, you're positioning the lens very near your eye and moving the specimen towards it to see magnified details. And the next is comparing in-grain surfaces. With a hand lens, the difference between in the fine texture in Eastern red cedar on the left in the top photo and the coarse texture in redwood on the right on the right in the top photo is evident at right and unknown specimen, and on the left also is compared to a known sample of Brahman on the right or 10 times. This one is the second photo of this comparing in-grain surfaces. This one, there's a lot of pores. The next is the reticle scales used to measure pore diameter. When a reticle placed over transverse surface and the sample is viewed with a hand lens, top pore diameter can be approximated, or division on the scale are 100 micrometers, and another type for reticle has bars of labid thickness above. This one in the photo is the reticle scales used to measure pore diameter. This one in the second one photo, this one. The reticle scales used to measure pore diameter are typically used in a microscopy to quantify the size of pores or openings in materials, such as filters, membranes, or geological samples. These reticles are either integrated into the eyepiece of a microscope or can be digital depending on the system used. And the next is the examination of microscopic features. The examination of the microscopic features. Preparation for examination of microscopic features involves a series of preliminary steps, including shaping the specimen, softening the wood, cutting section, and mounting the section on a slide. How well the slides are prepared will determine much information they provide. The examination of microscopic features involves the study of very small structures that are not visible to the naked eye, typically at the cellular or subcellular level. This process is commonly conducted using microscopes such as light microscope or the electron microscope and other specialized tools. The next is the shaping the specimen microscope. Work a piece of wood somewhere in a size between a sugar cube and a pack of gum in appropriate since it is large enough to be handled easily and small enough to be softened by boiling. Preparation of microscopic examination involves a selection of a portion of a wood that is free of abnormalities. Shaping the specimen is a crucial step in preparing samples for microscopic examination, as it ensures that the structures of interest is properly presented for analysis. The techniques used depend on the type of specimen or the biological tissue, inorganic material, and the microscope being used, or the light microscope and electron microscope. The next is the acetate gauge and millimeter scale. This clear acetate gauge, developed by the Forest Products Research Laboratory, contains lines of various widths that can be laid atop or alongside rays and pores to determine width and diameter shown actual size. These circles are useful for counting pores per unit area, just like in the picture. There's also, you can see also a rays in the up and pores in down, and circles in the circles in this picture. This next is preparing a sample for sectioning. A sample such as that shown above left need not initially have a radial and tangential faces to yield thin sections for microscopic slides, but it should be straight-grained as shown above right. First clean up the end grain, and then bevel the edges to leave a small portion or X from which transverse section can be shed. Transverse, as you can see, the transverse section can be sliced. The next is to find edges that can be beveled to produce radial and tangential faces for sectioning. The notches or stop cuts define the areas to be sectioned and making it easier to maintain the proper plane. The preparing a sample for sectioning is a critical step in a microscopy, especially when dealing with biological specimens, as it allows for the observation of internal structure at a fine level of structure's detail. The preparation process typically involves several key steps to ensure the sample is properly fixed and embedded and thin energy to be sliced into sections suitable for microscopic examination. And the next is the softening. In a few of a softer wood section can be cut directly from the sample block. Softening the sample by boiling. Boiling wood sample in water helps soften them for sectioning. Samples of average are sufficiently softened when they sink. About these pictures, or the softening the sample boiling is boiling a sample to soften. It is a process commonly used in various fields such as chemistry, material science, and food preparation. The basic idea behind this is that heat can break down the structure of a material, make it more pliable or easier to work with. And the next is the sectioning. Sectioning in a sample softened by water at room temperature, sectioning can be done when the surface is still flooded with water. Cutting with a double-edged razor blades. One edge of this double-edged blade has been taped for better control safety. The sectioning cut is made with a sliding motion. The arrow indicates the direction of blade motion in a full stroke. The cutting with a double-edged razor blade, just like in the picture, as you can see, there's also blades in here. Cutting with a double-edged razor blade is precise and efficient method commonly used in various applications ranging from crafting to medical procedures, just like in the picture. The next is the improvised slide rack. The improvised slide rack When making or examining slides, keep them organized on an improvised elevated rack, such as piece of scrap wood or even a book. The other hanging ends can be grabbed easily without touching the specimen area of the slide, just like in this picture. Okay, an improvised slide rack is a makeshift holder or organizer used for holding microscope slides, typically in laboratory or educational setting. It is useful when a proper slide rack is not available or when you need a temporary solution for organizing slides. The next is the mounting a slide. To make a temporary water mount slide top photo, arrange or one or more section. Just like in this picture, there are three types of this one. The mount slide top photo arrange one or more section in the central portion of the slide. This one in the first, and the second photo is to place a cover glass over a section. This one and the second photo. The third photo, as you can see, is with an eyedropper. Gently introduce water at the edge of the cover glass. Use enough water to surround the section. But not enough float the cover glass. As you can see, the mounting slide is the process of preparing a sample for viewing under microscope by placing it on a microscope slide and securing it with a cover lip. And the proper mounting ensures that the sample stays in place and provides clear focus images. And the next is the right amount of water on a water mount slide. When the correct amount of water used, it surrounds the section as is seen in the face view. The water forms a meniscus under the cover glass that holds the glass in place, flattens the section against the slide. Okay, just like this in the pictures, there are slide, cover glass, and meniscus, and in the down is you can see at the water and the section, just like this one. Okay, when preparing a water mount slide, the right amount of water is crucial for ensuring the sample is properly suspended, and that's it, and also to provide a clear view under the microscope. Too much water can cause the sample to float away or spill, while too little can cause it to dry out or become distorted. And the next is the too much water in a water mount slide, just like this one. It's just like they are the same, but this one is there are too much water on a water mount slide. Too much water leaves messy liquid edges along the cover glass. As shown in the face view, the cover glass is not held in place and the section is not held flat and level against the slide. When preparing this too much water on a water mount slide is having too much of water on a water mount slide can cause several issues that the quality of the observation. Okay, the next is tacking down the cover glass. Tacking down the cover glass is to save water mount slide for future use. tack down the cover glass with a gum labels top or cellophane tape above this one, as you can see in the picture. Taking down or placing the cover glass, or we can call it the cover glass or the cover slip, properly on a slide, it is a crucial step is preparing a sample for microscopic examination. A cover slip helps to protect or makakatulong ito sa pagprotekta about sa mga samples and to ensure the correct spacing between the sample and the objective lens, and minimize also the distortion or damage to the specimen. The next is the proximity of high power objective to cover glass. This one, as you can see in the picture, the proximity of high power objective to cover glass. With the slide mounted specimen is focused and the high power objective is extremely close to the cover glass, careless adjusting while focusing can shatter the cover glass against the objective lens, scratching its coating. To prevent this damage, locate the subject and focus it under a low power objective because rotating the high power objective into position. The proximity, as you can see this in the picture, these are the example of proximity of high power objective to cover glass. Okay, the proximity of the high power objective lens to the cover glass is an important factor in achieving a clear and focused image when using a microscope, especially at high magnifications. Typically, they are also, when you use a microscope, they are also 40 times, 60 times, or 100 times. As we can see the microscope, when you use it, we can see clear the pores of when we can use a wood to use a lens. The next is the evaluating hand-cut sections. While examining a hand-cut section for identification detail, you can also evaluate its quality and consider how you might improve your sectioning technique. Okay, when preparing a specimen for microscopic viewing, selecting the best portion of the section to observe is crucial. Obtaining a clear and representative image. The best portion or image, or we can call it the best portion, depends on several factors including the type of and the orientation of the section sample and the level of detail needed. The next is the best portion of a section for viewing. The best portion of a section for viewing are above are views of different portions of the same transverse section of the eastern white pine. The area along the edge of the section in the top photo provides the best view since it is thin enough to show cellular detail and it's relatively free of air bubbles. The section in the middle photo is also cleanly cut, but this area is thick and air bubbles obscure details. The section in the bottom photo is so thick with superimposed layers of cells that individual details are indistinguishable or the 100 times. The best portion of a section for viewing, and when preparing a specimen for microscopic viewing, selecting the best portion of the section to observe is crucial of obtaining a clear and representative image, or you can call it the best. The best portion depend on several factors, including the type of and the type of the level of detail needed, and the orientation of the section samples. The next is the sectioning problems. The damaged cell structures in the section at top left was caused by plowing rather than sliding the cutting blade, and the torn rug edges in the photo below it are from a dull blade. The ridge in the bottom photo indicate hesitation during the cutting stroke. The oblique view of tracked walls on the top photo at right indicates that this section is not truly transverse. Similarly, the apparently short rays in the photo below it reveal that this section is not truly radial or 100 times this one. As you can see, these are the example of sectioning problems. This is 5 photo. Okay, the sectioning problems are common challenges encountered when preparing specimens for microscopic analysis, especially what using techniques such as paraffin embedding or cryosectioning. These issues can affect the quality of the sections and ultimately the quality of microscope observations. The third is taking samples and keeping records. This one is the sample size is histology or microscopic examination refers to the amount of tissue or material used for sectioning and preparing slide for analysis. Prepare slides for analysis. The proper sample size is crucial for obtaining accurate and representative results. On approximately size sample ensures that the sectioning process is effective and that the specimen is well represented for microscopic study. And the next is the guidelines for taking samples. The guidelines for taking samples is taking appropriate samples for microscopic examination is essential to ensure that the specimen is representative of the tissue or material you wish to study. The guidelines for sample collection can vary depending on the type of study, just like the histology, the microbiology, the cytology, the specimen source, and the technique used. This one, okay. The next is taking sample from longitudinal edge of the object, just like this in the picture. This is the sample from nagaling sa longitudinal edge of an object, this one in the photo. To remove a small sample from edge of a board, first use a knife to notch in pair of stop cuts, just like in the A. This one, the A at the left. With the knife tip, score the edges of the sample area to guide the split B, just like this one. And finally, to engage the knife edge in the bottom of one or the stop cuts, and gently pry the sample free with a slight twisting motion of this knife, just like in the letter C in the down. Okay, taking sample from the longitudinal edge of the object refers to collecting a section or portion of the object along its lengthwise axis. The technique is often used to capture specific features of the objects that might not be evident in cross-sectional sampling. The next is taking a sample from end-grain surface. To remove a sample from end-grained surface, first use narrow chisel to make a pair of wedges just like this in the photo, as you can see, and also to shape parallel holes, or the letter A. Use a thin knife blade to connect the walls of the two holes and define the sample B. And finally, use also the narrow chisel to undercut and pry out the sample. This is the sample in the picture. Taking a sample from an end-grain surface refers to collecting a portion of a material, or the typically wood or similar fibrous material, from the end of the material where the grain runs perpendicular to the surface. This is after done to examine the internal structure. Grain pattern or other features that may be hidden in cross-section. When sampling from an end-grain surface, it is important to follow. Okay, the taking samples from historic objective. Taking samples from historic object is taking a sample from historic objects requires special care and attention due to their age, fragility, and cultural significance. The process must balance the need for analysis with the preservation of an object. And the next is the guidelines for keeping records. The guidelines for keeping records is keeping true and accurate records is essential in any scientific or historical or archival context, as it ensures proper documentation and organization and traceability. Okay, here we are about the wrapping and labeling a microsection. This section of microscopic examination can be wrapped as shown of safe storage. A self-stick label seals the packet and describe its contents. Okay, just like in the picture, as you can see, this is the example of wrapping and labeling a microsection. They need to label it, the microsection. Okay, the wrapping and labeling a microsection is an essential step in the preservation or identification and storage of tissue or material samples, especially the microscopy and histology. And proper wrapping or labeling ensure that the sample is protected, organized, and easily traceable for future examination or analysis. The next is the taking microsections from a table. The growth rings in the leg on the underside. This table make it possible to find the tangential face, which can then be cleaned with razor blade for direct sectioning. A or growth ring placement visible in the end grain of glue block B. suggests that a direct radial section can be taken along the edge of the block. Growth ring orientation in the glue block C, however, indicates the block would yield only tangential section directly, just like in the picture. There are also radial and tangential in here. Taking microsection from a table generally refers to the process of obtaining thin tissue slices or samples from a specimen using a microtome or other cutting tools, with an specimen typically being positioned on a flat surface such as a cutting table. A cutting table is often part of a laboratory setup used for preparing slides or specimen for microscopic examination. Okay, these are for today, and this is the end of my topic. Thank you.